I can get the wrong answer. So if I go to these studies in warfarin, for example, we know the clinical trial result is 2. In observational data, the hazard ratio that I get for warfarin on bleeding is 1. And I'm using standard adjustment methods that would be publishable in a typical journal. IPW, I adjusted for all the variables that are available. So I didn't do anything to make this bad. I didn't purposely make it bad. I actually did exactly what we would normally do. And statins as well. So we know that there should be some benefit, and I go study that question using standard adjustment methods, classic analyses that I think would be publishable, and I get the wrong answer. So the question then is the reason that the analysis is too confounded? Is Chris Granger right? Is it hopeless? So I can do the same thing, though. I can get the right answer, and you'll find out that it doesn't take that much. So in the same 2 data sets, using the same outcome, same adjustment variables, very similar data sources, and same definition of treatment, I can get results that agree pretty darn well with clinical trials. So on the right-hand side now, I'm seeing that warfarin elevates bleeding and statins are beneficial with respect to CV events. And so the question is, what changed? So to consider what changed, I'm just going to go through 5 of the primary principles of causal inference. The first question that we all have been talking about, that we immediately talk about with observational data, is no unmeasured confounding. We don't know that here. It's observational data. I don't know whether that holds, but I do know that I was able to achieve successful results with the same set of adjustment variables that matched clinical trials, so it might be plausible that we have no unmeasured confounding. And it's important to note that in the analysis that failed and the analysis that succeeded, the exact same adjustment variables were used. So it's not the acquisition of new variables. Then another primary principle that is done well in clinical trials is that the interventions and endpoints are well defined. And in the interest of time, I'll skip over that and claim that they are, but we could talk later. Then another topic is whether the measured confounders have been balanced well. So did the statistician do their job? In both cases, the result using standard adjustment methods and checks on balance, we would check standardized differences. Things are looking good. So propensity adjustment appears to work. We've balanced the measured covariates. So confounding does not explain the result. But a big difference in where the analyses depart is the new user design. So in the analysis that doesn't work out well, follow-up begins at the beginning of the data set, at a cross-section in people's lives, and they either are or are not on the drug. So they are prevalent users. And so when in the analysis that is successful, we use the new user design, where we're making sure that follow-up begins immediately after treatment initiation. So that's like a clinical trial. A clinical trial, when they give you 2 drugs, they definitely start following you up immediately. Whereas the prevalent user design, the one that failed, would be like a clinical trial where you waited 2 years and then started the analysis based on whatever they were currently taking. So you can imagine why that might have problems. The other question is to consider the topic of equipoise, which doesn't necessarily get considered in observational research, but has to get considered in randomized studies. In the setting, at least, of the warfarin example, I would say, and I'll talk about it later, that there probably was uncertainty and the patients were reasonably eligible for warfarin. But the analysis in Framingham pretty much purposely took all comers, and there were a lot of patients that were not reasonable candidates for statins. They were just available in the data. So as we have large clinical databases, it's very common at this point for me to see things that look like patients that look really different. So we're creating these cohorts out of observational data sets, and it might do well to consider clinical equipoise. Not that it has to be the same and defined in the same way as a trial, but that we can't totally neglect it. If we do analyses in patients that are not really reasonable candidates for treatment, no adjustment, no IPW, no balancing measured covariates is going to make the difference. So clinical trials have to meet all of these criteria, not just randomization. And I think that's really important. When we're arguing about observational research, whether it has value or whether clinical trials are better, we need to think more about not just randomization as the key difference, but of all of the ways in which a trial is rigorous and carefully done, and then question whether we can actually meet those same standards in observational research. And I will claim that often we can, and the future is bright, but we'll pause for discussion.
So we thought we'd break it up and have some question and answers in the middle here rather than waiting to the end. And I think one of the things that's always interesting in observational data sets is you look at every person who's in the data set typically and how the treatment's being used, whereas in a clinical trial, obviously you define that cohort by who's randomized. So you have this group in the observational data set who's not treated, but they aren't even included in the clinical trial other than being in the placebo group, for example. But they're pretty much the same patients as those who get the active treatment. So I think that's one of the biggest challenges, and it's always an issue of confounding, and that is most therapies used in the observational setting are used in healthier patients who don't have as many comorbidities. And so I think that's always a challenge of how you adjust for that or how you can control for that.
So Matt, I want to start with a question for you. One of the things you went through was you talked about the different principles that were important for trials. One of them was issues around sort of blinding and sort of who on the study team knew about exposures, et cetera. I guess the question related to that is, do you think those standards exist in observational studies or should exist there?
Well, I mean, you know who got the treatment, basically. They're getting the treatment in routine practice. So I think it's not possible to blind that in some setting. But we know that one treatment is related to many other treatments that a patient gets. So if they're in a clinical trial and they're getting a blinded treatment, their concomitant medications that may have an influence on their outcomes and the results, should be relatively balanced because the investigator or the treating physician doesn't know what other therapies they're getting. But I don't see how it's possible to really blind in an observational analysis, especially at the level of the patient or the treating physician. So I'm not sure if that's what you were specifically asking or whether the researchers who are doing the analysis should be blinded.
So that's exactly where I wanted to end up. So I'll switch to Laine here. So Laine's quite famous for a number of things. One of the things she's famous for is developing sort of a proposal for how to do statistical analysis plans and what sort of work can and should be done in the planning phase, sort of assessing, figuring out whether we should do the study or not. Do you think we should apply, or do you think we do apply those measures in observational studies across the board?
It's an interesting question, yeah. Because I do still probably adhere to that as an ideal, but I've certainly encountered why we don't always adhere to that. So the principle is, especially if you're using propensity methods, you can do the design phase first. You don't have to look at the outcomes. You can do the balancing, you can check the balance, you can look at who's at Equipoise, all the things I just talked about you can check first before you run outcomes. And you could say, just fix it, fix your design and separate, and then once you run the analysis of outcomes, you're not allowed to change it because that would be phishing. And so that's sort of the principle, if that's kind of what you're thinking along the lines, right? And so one of the challenges I've encountered with that is that oftentimes we are hopeful that we've got a good analysis, and then when we run the outcome model and it looks totally wrong, we come to Jesus and remember that there were confounders we could have, should have measured, and then we're going to pay to go get them. And sometimes they're not all just sitting in your data set, available, ready, easy. And so sometimes you really do improve your analysis in that second round. Like usually when I see us going back to redo something, it's because we realize we can do it better. And so to stop that or to be rigid about that has a downside too. So I'm still a fan of the principle, but I don't always execute it.
Excellent. No, I appreciate that. So during your talk, I got an anonymous text from a DCRI MD, and they asked me, is 1.49 the same as 2?
Oh, look at the confidence interval.
That's my response. So Matt, I know you're anxious to get to the second session, and maybe we should start with that. And then, but at the end, I do want to ask, it seems like some of your work is actually crossing over trials, observational studies, et cetera. Do you view that as the future of what we would be doing at DCRI?
No, I think we can generate evidence from a variety of different sources, and many people have done that and been fortunate to work in all those different areas. And I think when you look at it for a given treatment, for example, taking the clinical trial data, looking at that in different populations, not only from the United States, but from other countries as well, is important to understand what the treatment effect is and how it's used in practice and what the outcome may ultimately be. So I think they're iterative, and evidence can be generated from multiple different things, as Laine said, even with the series in the New England Journal this year. So I think there's a room for both, but you have to understand the strengths and limitations of both. And we do numerous secondary analyses from clinical trials that are fairly well known, most of which are often subgroup analyses looking at the randomized treatment in different subgroups. And many would argue those are inappropriate, but I think they're hypothesis generating for future work and trying to understand that. So I think understanding the goals and objectives of what you're doing and defining it ahead of time can limit some of the bias and actually help you then not overinterpret your findings.
Okay, thank you, Matt. One other comment. So Laine, you obviously do a ton of work both on the methodology side as well as the applied work. So in terms of the trials going on and outcomes, if you're doing a cluster randomized trial, where do you see that fitting in the spectrum of sort of the pure randomized trial versus more observational study, at least in appropriateness for the methods?
You'll have to answer that question. I haven't done a single cluster randomized trial.
At least my impression is you end up doing a randomized trial that has almost exactly the same issues data-wise that an observational study has. So.
Okay, we can talk about that in the second part. Okay. Any questions at this point?
So of course I have to comment, right? I mean,
Yeah.
but Laine, so I love the idea. I really do like this idea of taking treatments where we have a very kind of narrow confidence interval about the known effect, and then using those to see how well or how well we may or may not be able to replicate those in observational data sets. I really like that. Of course, we have lots of examples. The challenge, I agree with what you've been saying, but the challenge is because sometimes you can come up with a observation that approximates what we know to be the truth based on the only way to really account for measured and unmeasured confounders, doesn't mean that that's the right thing to do, because we have lots of examples where that's not been the case. So many examples. Back from hormone replacement therapy to erythropoietin.
I have a slide on that.
We have so many examples where we know we can't depend on these types of analyses, and in fact they result in substantial public health harm that I think we need to, we can't, because we can be successful every once in a while doesn't mean that we should be doing it.
Yeah, so you bring up a great point, which is that examples don't make a rule. And so no way should you say these examples mean if you copy this analysis, it will always work. What does exist is that all the 5 principles have been proven based on theory. They're known to have theoretical justification. Without them, you can't have causation. And so what is interesting to see is that if you adhere to the theoretical principles, indeed we can replicate reasonable results. One could argue whether they should be identical because the populations are different. And that we don't necessarily have to be so discouraged that observational research has no future or is always wrong, because indeed when we can adhere to the principles, it is actually possible to see results are exactly what we should see. And so it's an example of why the principles matter, but I would agree that the examples are not the rule.
But I would also point out, Chris, that those examples you used were examples that use traditional adjustment at baseline.
I have a slide coming.
Okay, great. So
That's fine, but if I can just...
No, you can't. It's mine. Sorry.
Sorry, but the key is that you can't know whether there are unmeasured confounders until you have another gold standard to test that against. You
So
can't
I
know
think
that.
we're using the word truth a little fast and loose here. If you are recruiting half a patient per month and you see 100 patients a month, and you are one of hundreds of providers who see those types of patients, this is a huge selection bias, which can also have public health consequences when you apply the answer, not the truth, but the answer that you got in this incredibly select, highly controlled situation to the rest of life, which doesn't work that way. So this is why Matt said it's iterative. You have to look. So we've had efficacy studies that weren't effective, and so you can't really say truth.
Well, that's fine, but Matt's going to address this. Then the answer there is not to forego randomization as the only way we can come up with equal groups at baseline. It's to do trials that are more generalizable because we are less selective. I think that's where we want to get to.
But that assumes then that you can always do a trial. I mean, given the number of conditions or situations for which you're not going to do a trial, we do also want to be able to do observational research in a way that mirrors as many advantages of a trial that we can. I mean, there's a lot of good things about a trial, and we can emulate more of them.
That's a good segue, I think, into part 2.
Cool.
Yeah, let's do that. Okay. All right, well, that's good. We wanted to get a little bit of a discussion in the middle, which was exactly what we had hoped for. So, real-world evidence, I think everybody here knows what that is. There are huge sources of data available now that we can get from routine practice, both from the electronic health record, from claims, and other registries. And the key now is we have common standards for how those data are developed and how they're archived and how they're analyzed, so that this is a data source now to do a new type of clinical trial, pragmatic trials. And so I think we know, and we've talked about traditional trials on the left here, and that is it's a very select population, it's a narrow investigator pool, it's controlled with blinding and placebo, and you have a standalone data collection. Whereas with pragmatic trials, you look at a routine population, we hope. You have a larger pool of investigators or just routine clinicians taking care of patients, typically done in just a single or a few countries, typically done in an unblinded fashion because it's often too difficult to do blinding and storage of a placebo or blinded drug in a pragmatic setting. And so we often then would have an active control treatment, and we have centralized data collection. But the thing that links this is randomization. I think that's what Chris was making that point is, even in a pragmatic trial, there's still randomization such that the cohorts of patients who are studied should be relatively equal. And these are a series of principles proposed for pragmatic trials that are different but similar to those proposed that I talked about earlier for traditional trials. And that is that patients are randomized or stayed on the randomized treatment as much as possible. We have reasonable ascertainment of outcomes and other things. And I'm not going to go through the whole list, but as we think about the evolution of clinical trials, we need to think about quality and how that is defined and then determined when a pragmatic trial is done. So ADAPTABLE, I think all of you have heard about this. We've talked about it at previous research forums. It's the first intervention trial done out of Pragmatic, where we're studying low versus high dose of aspirin in patients with chronic cardiovascular disease. It's an over-the-counter therapy. It's an open-label study where participants are self-randomized through a web portal and then followed up through phone and web portal contact, as well as queries through the common data model with Pragmatic and other data sources. And so this trial is trying to answer the question of which dose of aspirin is more effective and safer for patients with chronic cardiovascular disease. And we have enrollment rates from ADAPTABLE that are quite different than what you see with traditional trials. We have some sites, including our own at Duke, that are enrolling up to 60 patients per month at a site. So you'd think this is still a much more representative population than the traditional trial that has half a patient per site per month. But only 4% of the approached participants actually agree to enroll and randomize in the trial. So we're approaching hundreds of thousands of patients to get the patients enrolled in the study. So there still is a selection bias that's unavoidable, I think, even in a pragmatic trial. But informed consent is necessary, and you have to recruit patients, and they have to consent voluntarily to participate in the study. This is a trial that was just presented a few months ago from Sweden, conducted in the country of Sweden, where they were looking at patients who were undergoing percutaneous intervention and studying unfractionated heparin and bivalirudin intravenous anticoagulants. And the key here is that almost all of the centers that did this procedure in Sweden participated in the study. Almost half of the patients with the disease of interest were actually enrolled in the trial during the time period that it was conducted, as shown here. And if you look more narrowly at the pool that was eligible for enrollment, 70% were randomized. So that's a much more representative population. But when we look at the groups who were randomized, shown in red and blue, compared to the group that was screened but not randomized, there are clear differences. And so even though the randomized groups were similar and comparable, the group that's not randomized does have higher risk features. And so they're not the ones who the therapy is being tested in. And so I think we need to recognize that in pragmatic trials, we still don't look at the whole population and understand fully how these treatments may work in patients who are not eligible for the study or who cannot be consented for a variety of reasons. And so randomization is the critical factor that's still present in pragmatic trials. When you don't have blinding of the treatment and other things that go with a traditional trial, you may introduce bias, and that remains to be seen. And that may influence how you ascertain outcomes, how the patients are retained in the trial and stay on therapy if it's a longer-term therapy, like we're seeing in ADAPTABLE. But we can recruit larger groups of patients that are more diverse from a larger pool of investigators and sites, however you want to call a site. And so there likely will be the synergism between traditional trials and pragmatic trials when you're looking at a given therapy and its development cycle of how do you assess the treatment effect of a therapy early on in its development when it's going for an approved base indication, and then later when it's being used more commonly in routine practice. And so I think the pragmatic trial is the bridge between the traditional trial and the observational treatment effects. And next, Laine will talk about some newer approaches and new methodology that's being used for comparative effectiveness treatments.
So I think a nice bridge, and I want to pause too because people are interested. So my intention was to skip the top 3 principles, unmeasured confounding, definition of endpoints, and adjustment techniques like propensity score methods, not because they're not important, because it seems like they've been widely talked about. Like if you came to a talk on observational studies, you expected to hear that. And so I wanted to talk about some other aspects, but I will pause for a minute, particularly because of Chris's interest in the no unmeasured confounding assumption. I think an interesting point, a case in point, is that the study I'm working on uterine fibroids, where there's no way we're going to randomize hysterectomy, in order to make that actual plausible that we might have no unmeasured confounding, we're doing a lot that's never been done before in another registry in terms of going and getting the imaging and getting the detailed fibroid features, the weight, the dimensions of every fibroid. It's a pain, it's expensive, and it's one of the burdens of the study. But the study's bothering to do that because they want to do causal inference. And so we can also sometimes, when we know we're not going to be able to randomize, do a better job getting those confounders so it's plausible. So I would in no way minimize the point you're making as to how important measuring confounders is if you have to do observational research. It's like the main thing I talked about for a whole 2 years on that study. But I'm skipping it today only because I feel like that's something people really well or have a lot of knowledge about. So I wanted to focus on the 2 things that differed in the 2 analyses that I saw, which was the concept of new user designs and Equipoise. In particular, too, because that's an area where there's some new methodology that we can be considering. So the prevalent user design, just to clarify what we're talking about, I'm picturing the anticoagulation example, and so calendar time is my scale across the bottom, and this might be kind of the profiles of patients. And so treatment might be the time that somebody starts anticoagulation if they have atrial fibrillation, the TX, and they go along, and at some point they might have a bleed. And if they have a bleed on anticoagulation, for the purpose of exaggeration in the example, I have said they are taken off anticoagulation. So as we see them later, the line that's vertical would be the start of a registry or the start of a clinical trial. This happens in Aristotle, the clinical trial in Orbit. Anytime we have a start point and our data is there at baseline, we're going to see their treatment status. And in my exaggerated example, what kind of patients are left? The kind of patients who are left still treated at our registry start time are the type who don't bleed. That's the reason why the prevalent user design tends to give the wrong answer. So we could assume that there should be selection bias, and we should expect to see an attenuated toward the null result for the risk of warfarin on bleeding due to the fact that many of the patients who may have bled would be selected off the treatment or out of the sample by the time we see them. And so selection bias, this bias of selecting out of our sample, is not addressed by our typical confounders. And that's the most important thing, because I wanted to say today is that so many people think statisticians do adjustment. We have selection bias. Laine, go do adjustment. And I have some variables, and I'm going to run a propensity model, and it's going to be balanced, and you'll be like, good, it worked. The methods we use and the adjustment variables we have do not adjust for this kind of bias. So it is not getting addressed. The kind of variables we would need to adjust for selection bias would not be the usual confounders. It would be all unobserved causes of bleeding. Everything in the biology. Not just the stuff we see. We make treatment decisions based on confounders. But the biology involved in selection bias is much more complex, and we don't have the adjustment for this problem. So this is addressed by design. And I have the famous nurses study, because I'm not the only person saying this. Miguel Hernan's going around giving a similar talk. And the famous result with the nurses' health study is that hormone replacement therapy appeared to be beneficial in the old analyses. The observational data sets had been showing that it was beneficial to be on hormone replacement therapy with respect to coronary heart disease. And then the Women's Health Initiative randomized trial showed the exact opposite, that it was harmful with a hazard ratio of 1.6. Then Miguel Hernan did the analysis in the same data set with the same adjustment variables, but just restructured to a new user design, which is not a trivial analysis to do. For those here that have supported me in doing it, it's a bit of work. But using a principled approach to identifying new users, the same observational data that had been giving the hazard ratio of 0.67 gave a hazard ratio of 1.2 in line with the clinical trial result. So it wasn't inherent in the data that you couldn't get a good result or that the analysis was biased. In this particular case, it looks largely attributable to the prevalent user problem. And he gives a much longer talk on the details around that, including a good paper. So if we know we should use the new user design, why are we not always doing it? The new user design sometimes has too few patients, so we've certainly encountered that the number of patients using things at baseline is much more than the number of people I can have identified during follow-up, and that in order for it to be unbiased, adjustment variables need to be collected longitudinally. So we need to know why somebody's starting longitudinally, and that's sometimes not feasible. So depending on the data set, it can be done well or it can be done poorly. But the solutions that I think why it's more likely to become possible is as we have larger quality improvement registries and larger clinical databases, which I'm certainly seeing in outcomes, we have much more opportunity to get a decent sample size. So we don't have, the sample size restriction doesn't have to be as big of a deal as it used to in, say, a smaller registry. And then a lot of my projects have started to collect a lot more information on longitudinal follow-up. So the CHAMP-HF registry has detailed information about symptoms and treatment changes with dates and all that kind of stuff that I would need to be able to adjust. So as we're improving our ability, pulling this information in, I think in the future as well from EHR and claims so that we don't have to get somebody to fill out a CRF with all of it, the possibilities in the future to have enough sample size and have enough information to do new user designs, I think is increasing. And so then we also have to have methodological improvements to help promote these methods and help do this work. And so there's a couple methods called sequential stratification, risk set matching, dynamic matching. Everybody who does a method writes their own name for it. So it's really confusing in terms of jargon. But they all really come down to something along these lines, just to give you the intuition that when a person starts treatment, you have to have a relevant time scale for this matching to occur, but a great time scale is time since eligibility for that treatment. When somebody starts treatment, you can go acquire another person who looked just like them, who did not start treatment, and follow them from that time forward. If you create a bunch of these pairs, pull them together, they're a new user design, and one can pool the results over that. So there's hundreds of papers in this area, and there's a lot of development still going forward. There's a lot of open questions methodologically, but these are the types of methods that were used by Miguel Hernan in the Nurses' Health Study and that I used to get new users. So also, I've done a couple papers, and I just wanted to thank Orbit and Aristotle for supporting these methods because they're slow and time-consuming. And they shouldn't be forever, but they are now because I'm still learning. So another topic is to consider, do we care about equipoise in observational research? We know we care about it in a randomized trial because we're not allowed to randomize if we don't have it. But maybe we shouldn't totally throw it out the window in observational research, kind of at least keep an eye. So these are distributions of propensity scores, or the probability of being treated, and this is something that's really facilitated by taking a propensity approach to your analysis. So if you take a propensity approach, you can look at the propensity score distribution among the treated and untreated. This is simulated data, but both examples come from real things I've seen. On the left, it's a TAVR-SAVR intervention for heart valves, and on the right, it's flipped, but OAC. So these are examples that I really see, where you can see on the left, among the treated patients, that's the dark line, tons of people have a propensity score of almost 1, which means their chance, at least in the current practice in that data set, physicians are in agreement that type of person absolutely must get treated, 100% chance, or nearly. And on the other side, among the untreated, there is tremendous agreement among everyone on certain types of patients, they absolutely will not get treated. And so where is equipoise? Hard to say, but maybe 100% agreement among practicing physicians that nobody of that type should get treated might suggest we're not in a position of equipoise. And one of the reasons, well, a couple things. So who would we consider putting in a clinical trial? In these pictures, I don't know exactly what threshold we would pick. but maybe somewhere in the middle. And so I drew some lines to suggest that might be who you'd be willing to randomize. Now, the propensity is not the thing on which we define equipoise for a trial. So this is just connecting to the concept, but it's not literally. We think about patient characteristics in defining equipoise, not propensity scores. But propensity scores could give us a warning that we're way out of the ballpark. If you have ones and zeros, that person is somebody for whom everyone in current practice thinks they know the answer. So maybe they do. So the problem is, we of course want to use observational data to extend generalizability. And that goal is at tension with the concept of equipoise. But we can think about over what dimension are we extending generalizability. Maybe we want to be more geographically representative, socioeconomic status representative, a whole lot of things. And of course, even clinical risk factors. Not all of that is what we have here. How broad do we really want to go? Do we really want to try to answer causal questions in people for whom practitioners apparently know the answer? Maybe that's the purpose of a study. Maybe we don't trust current practice, and we actually do want to test causal questions in patients for whom everybody already thinks they know the answer. But it's one thing to want that. It's another thing to realize that there's very little evidence in those tails. So regardless of whether we're interested in those patients, they have high variability and the potential for high bias. It's kind of like a randomized, if we were randomizing them, we would be using a coin with a chance of 1% to get treatment, or near 0.01. So imagine a randomized study that gave 99.9% of patients treatment A and 0.01% of patients treatment B. That would be a really inefficient randomized study. That's sort of what's happening here in the best case scenario. Given I can adjust for everything, no unmeasured confounding, oh, and my randomization coin is terrible. So if we consider the propensity approach as a possible way to get red flags, we might want to reconsider maybe there are other things, maybe there are people for whom we don't want to stretch that far. So there's a trade-off when we ask observational data to answer harder questions. When we ask it to say, be more generalizable, tell me about everybody who I have in this data set, where the data is sparse and the biases can be strong. So I wouldn't say that we don't ask stretch questions when they're needed. I think we still should. That's one of the advantages of observational data. But we should possibly check how far we're stretching to answer something that is poorly informed by the available data set. And the way we can do that methodologically, one approach is to check the propensity distribution and take a step back when you see stuff like this, and think about who possibly is not, maybe somebody's in your data set you didn't mean to have there. Why are you studying people who everyone knows how to treat? Possibly refine the population scientifically, but it actually does happen a lot that I've noticed that we can't. So we think we've got the population and we still got a distribution like that, in which case there's other methods that are coming. For example, overlap weighting is a method that was developed by Family in the Department of Statistical Sciences here, and she and I both really like it. And that weighting method reweights the population towards the center. So where the original population had these two tails that we can see on the left, the green line, I don't know how well you can see it, is the distribution of patients after overlap weighting. They get pulled towards equipoise automatically. So if you can't decide, you don't know what it is about these patients that are making them so extreme, you don't want to exclude anyone, the advantage of this is that it adds efficiency, it stops having the problem where the tails add all that inefficiency, pulls toward the center, and can be a methodological approach if there's not a scientific one. So my feeling is there's so many reasons to do observational research, but we can design rather than simply rely on the disclaimer. This disclaimer that I write on every paper, as with all of observational treatment comparisons, we cannot rule out the possibility that associations are biased by unmeasured confounding. I think that's an appropriately cautious statement, but it would be a shame if that statement eclipses the potential to do good causal inference in observational data. And I think we can do even better observational research by appreciating the strength of clinical trials. That's my end.
Thank you for round two. So do you have any comments on Laine's presentation? It seems like the two of you were actually coming together at the end. She's trying to emulate your methods, and you're talking about hybrid approaches.
No, I think that's the case, and I think that part of it is you're trying to apply this ahead of time. Again, not getting into the analysis and then trying to take a different direction, but looking at the strength of the data ahead of time and then applying these new methods, which again are worthwhile talking about. I think in this setting, there's value to different types of analyses, and what we want to do is show the comparison and contrast here. There always will be unmeasured confounding, and we can't get that because we never really know how physicians are making decisions to use treatments in their patients. But assessing that in the larger population and with the methods that you use, I think adds value and adds strength to the evidence of what the treatment effects are.
Yeah, no, I agree that it was very much by design that we converged, because I think that some of the strengths of observational research are what is being emulated in pragmatic trials, and whenever you can randomize, nobody would dispute the value of it. But I have enough projects where that's not going to happen, and so we're trying to see like how can we become even more like a pragmatic trial. Basically, we might be able to do pragmatic observational studies that converge to that same goal.
Yeah, and I think these data sources allow for that to happen. So you can do the trial and the observational analyses in the same data source as the foundation. And that's, I think, one of the strengths going forward is that you have a common data source, and then that's used for both purposes. And you can even do them in parallel, for example, in that regard. If you're doing a randomized trial and then looking at a treatment effect in patients who are not included in the trial, that also may be an opportunity going forward with millions and millions of patients in some of these data sets.
Yeah.
Excellent. So I did want to make 2 or 3 comments and then maybe take questions from the group. So one of the things that's come up with observational studies is we talk about how it's more generalizable than a randomized trial, and we sort of take that for granted. I do want to make a specific comment about that. So many of my projects are in the heart failure network, and in the heart failure network we've done a dozen or so trials, and then in addition we do a bunch of secondary manuscripts off that. One of the cases I had, and I put together a few slides, I don't necessarily want to go through them now, but there's a statement, and Rob Mentz will laugh at this statement, but we basically looked at acute heart failure patients from this trial, this trial, and this trial, and the trials were Dose, Caress, and Rose. And that means something to you, probably not to the rest of the group. But what I think we're trying to say as like investigators is we studied acute heart failure. It seems like a general statement. But when you put together the inclusions and exclusions from that trial, I think there were 18 inclusions to get in those trials. There were like 65 exclusions to get out of those trials. Then you end up making statements later in the paper where you say something like the unadjusted rate of whatever it is some number. And I actually put together some SAS code. I guess I'm interviewing for a SAS programming job later. But one of the things I said is what you did versus what you meant. So what you did was you took a hyper distorted population, like with a massive astigmatism. You estimated something. Your intention was actually to do something else, and that is basically to estimate something in acute heart failure. When people actually take your results, they're going to take your results as if you took it from a general registry of acute heart failure patients. So I think that's a huge potential problem. That's sort of one problem. I don't know if that's for or against observational studies. Another problem, and this kind of gets at what Laine was talking about with the sample size things. Many of the areas I'm involved with, we care about things like time to event, like clinical endpoints, and we have sort of a relative, like, would you believe an effect of this size? And oftentimes it becomes almost implausible to believe something where you're going to have a hazard ratio below, say, something like 0.75. Like, so if I say I have a new treatment or this treatment exists and it's going to make your hazard ratio, instead of where it is, 0.5, that's almost implausible. But when you sort of take those sort of beliefs and you sort of say, how many events do I need? Oftentimes you become sort of out of the clinical trial business. You're sort of priced out. So that's sort of another problem. Another thing related to that is the cycle of how long things take. Many, many years ago, a lot of people here were involved in a study using the Duke Data Bank, which I think was one of the beautiful observational studies. And one of the things that was so nice about it is that so much of what happened sort of at a very specific point in time. And so a lot of the biases you're referring to were sort of, we didn't have to pay attention to them because they were kind of automatically washed away. But we basically published a paper, and in that paper at the end, we said something like, we would recommend that you do a study of 10,000 patients. We sort of described, roughly speaking, how you'd power it, et cetera. That study was done by a different group. It was funded by something like 8 different companies. That result didn't come out for like 9 more years. And so the cycle of how quick we can do things is actually, I would say it's incredibly slow.
Yeah.
So I just want to make those brief comments.
No, thank you. It was good summary. I mean, there clearly are, I mean, despite all your best efforts, there clearly are limits with the randomized trials and time and biases and so forth.
Can I ask a question with ADAPTABLE? Why do we think the sort of approached participation rate is as low as it is? I was surprised by that number.
Well, I think the key is that we don't really know the right approach now to try to engage participants in the study. Many of them are being recruited remotely by email or by letter, and they're not having a direct conversation with the provider. So they have to be contacted multiple times in a row in order for them to be randomized in the study. And we're still learning why they would choose to participate and why they would not be. Most people are already on aspirin, and they may choose if they don't want to have the chance of changing their dose. And so, that's one of the things that's an unknown right now, of how many patients you have to try to approach or screen to then randomize into pragmatic studies. And in this case, I think you have the bias that most of them are already on the treatment, and they have to agree that there might be a chance that their dose could be changed.
Okay. So I'll take a question if there is one. I do want to make one comment. I think we are working at the right place at the right time, and that we have incredibly detailed data sets to be able to do some of the stuff that Laine was talking about. So, Eric?
Matt, just to address your last one, the issue comes in the study that many of those patients weren't at Equipoise, right?
Yeah.
The clinician and the patient weren't at Equipoise because the patient, for whatever reason, really wasn't a candidate for adaptable. We see them all the time in our clinic. You have the ability to actually collect, if you would, a question just a simple question of asking us, would this be somebody you really think should have been randomized in adaptable? If you had that, and we also have all the clinical data, then actually a lot of what Laine is talking about could be done. You could look at the overall population. You could look at a population amongst for whom the question was really relevant. The patient was at Equipoise to actually be randomized, and then see prospectively whether the answer was the same. The other thing I might challenge Laine is she will have the answer before you actually have the results from the trial. So you should have Laine now do the results and see how close she is. Because I really had this huge problem with the results of observational studies trying to see how close they can replicate randomized trials. Because in fact, you know what answer you're trying to hit for, and lo and behold, you can find methods that'll get you there, and you can find methods that'll take you farther away from that, but you ignore those because you wouldn't report a result that's farther away from the result that you actually wanted. The final
I
thing
don't
is,
have time to run that many
No, I got it.
analyses.
Yeah, no,
I'm not-
great
always-
points,
This is
great
real.
points.
but the other final thing is, it's kind of funny, Kevin, the choice you chose. Unless I'm mistaken, the example we found from the data bank, which actually did change practice, actually wasn't defended by the trial itself. When the trial was done 8 years later, the result was neutral, that there wasn't harm seen. So in essence, it's an interesting example to choose, right? Should our study, although it was profound, have changed practice?
All great questions. More work to do. I think that's a nice summary of the things we're trying to talk about today. So it's hard to summarize better than that, in my perspective.
Yeah, plus our time's up.
Yeah. All right. Thank you, everyone.
Thank you all.